The human body is made of trillions of different cells, and each of them contain exactly the same DNA molecule and the same genetic information in their nucleus. That's quite interesting concept, as cells can look very differently and they actually can do very different jobs. Lymphocyte, it is very different from an hepatocyte or a neural cells, but they do contain and they are led by the same genetic information. What does it make those cells different from each other, starting from the same direction contained in their DNA? And what it is the subset of genes that each of them express. So they have all the same genes, but only a portion of the genome is expressed. Which set of genes each cell needs to express is regulated by a large number of different factors. During development of an organism, position factors play a really important role, hormonal factor, growth factors, the vicinity of other genes, and of course, an important player is the environment. And in the environment, the diet always has an important role. We have been studying the ability of nutrients and food components to regulate gene expression since the late 80s. So this is not a new thing. And we have accumulated over the year a lot of information. For example, vitamin D or other liposoluble vitamins can freely get inside cells through biological membranes because they are actually soluble in lipids. So it can go straight into the cytoplasm where it binds a specific cellular receptor, but the vitamin D receptor, that it goes into the nucleus and activate the expression of calcium transporter. And that's one of the mechanisms how vitamin D affect calcium absorption in the intestine. Also, iron regulates the expression of genes that are involved in its metabolism directly, and that's a post-transcriptional level. So iron, through the binding with an iron binding protein, a regulative iron binding protein, is able to regulate the stability of a messenger and the efficiency of translation. So we are able now to investigate the effect on global scale of a given stimuli, such vitamin D action through its receptor. So while we used to test one or few genes at the time, now we are in the position of test the effect, for example, of vitamin D on the expression of the global, what we call now transcriptome, all the genes at the same time, and that allow to understand that the effect of vitamin D is also in other tissues that were not classically known as affected by the availability of this vitamin. For example, a nice experiment was done in skin, and we now know that through vitamin D receptor, vitamin D affect and modulate the expression of a large number of genes. And interestingly, the pathway that were identified as modulated by vitamin D and its receptor are involved in cell proliferation and differentiation, and then can suggest that vitamin D availability can have an effect on the development of cancer, skin cancer, for example. So the effort to complete the Human Genome Project was a big incentive to develop a really efficient new technology to sequence DNA and to analyze big complex amount of data with new developed bioinformatics technique. This is on the one side is very exciting because we are uncovering mechanism of interaction that we had no idea before, but also highlight how complex is the system. So nutrients and food components are able to affect the physiology of cells in a way that we had no idea before.